Next speaker is Dr. Nancy Jones. She's the Vice President of Clinical Development at Neuren Pharmaceuticals, and she's going to talk to us about their clinical trial for NNZ 2591 as a treatment for Angelman syndrome.
Thank you, Allison. Good afternoon, everyone, and greetings to all of the people who are virtually joining us. We haven't forgotten about you. We're very happy to be able to be here today to give you an update about our program. Some of you have heard about our program before. You've been here the previous years, and unfortunately, this is our first time back in person since COVID, and it's very great to be back and see the energy in the room. But there are probably many new people, so today I'm going to give you a little bit of an introduction of Neuren, as well as our milestones that we've met into the program that we've completed so far, and then where we're at currently. So Neuren Pharmaceuticals is a specialty biopharmaceutical company. We are specifically focused on developing treatments for neurodevelopmental disorders. We're a small company. We're based in Australia, and we've got a small group of people internationally. But as a group, we are very personally and professionally passionate about developing treatments for individuals that have rare genetic disorders for which they have great unmet medical need, but there currently isn't any treatment available that is specific to the disorder. So as you can see in the table, we have two drugs in development. One is called trofinetide, and then there's NNZ 2591. It doesn't yet have a snappy name. I did want to mention something about our trofinetide program. You can see on the top, it's being developed for Rett. We have actually completed a phase 3 pivotal trial, which had a positive result, and that is with our partner in North America at Pharmaceuticals. And it has been submitted to the FDA for review for an approval for marketing in the United States, and that is scheduled for March of next year. And I mentioned that just because I was reflecting, when we started in Rett syndrome, it was 3 sites, and we were the only sponsor in the field at the time. Now there are more, but we did have very passionate clinicians who were dedicated, as well as a very robust natural history study. So I mentioned that in terms of encouragement, and as a reminder that we've seen a lot of steps, lots of things in early phase 1, phase 2. Even with sort of small beginnings, you can meet sort of great milestones. So hopefully it's still not yet a done deal. It still needs to be approved, but it's at the point where it's with the FDA for review. NNZ 2591 is the compound that I'll talk about today. It's in development for 4 rare neurodevelopmental disorders, and this is the drug that we are actually developing for Angelman syndrome. So where does it fit in terms of the drugs that are being developed? You've heard a lot today about new drugs, and then you can also repurpose existing drugs or supplements that people use. You can do gene therapies, you can do neurobiological treatments, which are treating the things that are the consequences of the genetic disorder, and you have treatments for specific symptoms. And NNZ 2591, which is different from the other drugs that we've been talking about today, is a neurobiological therapy. So what is it? So I'll read this and I'll unpack it for you. It's a novel synthetic analog of a metabolite of insulin-like growth factor 1, which is called cyclic glycine proline. So IGF-1, insulin-like growth factor, is a growth hormone in the body. It's produced throughout the body, including the brain, and it's responsible for the growth and development of the neurons, as well as the supporting cells that help maintain the neurons, including the microglia. And when it acts naturally in the body, it breaks up into smaller pieces, and those smaller pieces are called metabolites. So one of those metabolites is called cyclic glycine proline. NNZ 2591 is a synthetic analog of the naturally occurring molecule, CGP. And what that means is it has exactly the same molecular structure as the naturally occurring molecule, but there is one small, slight chemical change that you make, and that makes the drug more amenable to be used as a medication. So it means that the drug will act exactly the same way as the naturally occurring drug, but you can then use it as a medication. So you can see in the slide on the right, this is the properties of the medication. It's orally available oral. It's a liquid formulation, so you can take it by mouth. It has 100% oral bioavailability, which means that all of it's able to be used and absorbed. It crosses the blood-brain barrier, and then it has the good pharmacokinetic properties, which again, as was mentioned before, is how the drug is used and absorbed by the body. So this is a snapshot of where we're at in terms of that drug development process. And the items in green are things that we've completed, and the item in purple is something that is ongoing. So just highlighting some of the preclinical work as this relates to what does NNZ-251 do on the cellular level, and why is it relevant to Angelman syndrome. So what we've seen from our preclinical studies is that it can improve the function of certain brain cells. And I mentioned earlier there's a brain cell called a microglia. And that's a brain cell that helps support the neurons, the main cells that talk to each other, and it helps maintain the structure and the connectivity between those neurons. In treatment, in preclinical studies, NNZ-2591 is able to improve abnormal function. That in turn also is able to help rescue abnormal morphology and density of certain parts of the neuron. So the dendritic cells, spines, are those parts of the neuron that reach out and receive the messages from other neurons. And it's been observed in Angelman, they can have abnormal shapes, morphology, as well as abnormal density, so the number of them. And that gets rescued with NNZ-2591. Additionally, then you get improvement in impaired signaling between the cells, which is also something that is observed to be synaptic strength and abnormal in Angelman. And then it also improves the bioavailability of insulin-like growth factor 1, or IGF-1. So in terms of further evidence of support for why is it relevant for Angelman syndrome, we have done an animal model study, which was a 6-week treatment, which fully rescued the phenotype in the mouse model, as well, including seizures. And what you're seeing in the slide are all of the behavioral measures that were conducted, and it covers from a range of analogous symptoms, such as anxiety hypoactivity, activities of daily living, or sort of functional activities, sociability, motor, and cognition. And what you're seeing in each little cell is on the left is the wild-type mouse that doesn't have an impairment in the darker color. And you can see in each of those cells on the left-hand side, the wild-type mouse is really quite different in terms of the performance from the Angelman mouse. And on the right is what happens after treatment, where you can see all the bars are equal to each other, meaning that the Angelman mouse has had their behavior rescued or improved. And then the wild-type mouse has not had any negative effects. So their regular performance hasn't been sort of negatively or deleteriously affected. So in terms of clinical trials, we have completed a phase 1 study. We had a phase 1 study in the healthy population, so healthy volunteers who were adults. It included a single dose stage, and then also a multiple ascending dose stage, where they received twice daily dosing for 7 days, and they either received 2591 or placebo. So there was a placebo comparison for the safety data. And overall, the safety profile from this study was very good. The adverse events were all mild or moderate and resolved during the study. There were no serious adverse events or severe adverse events, and then there was no clinically significant findings on any of the lab values, vital signs, cardiac tests. There was one discontinuation in the study, and that the person's AEs resolved once they stopped taking the medication. And the most common adverse event was drowsiness. In terms of PK, basically what we saw was a predictable PK profile, which is what you want to see. You want to make sure that you have a sort of a systematic way that you're seeing the drug is being uptaken in the body. So where we're at now is at phase 2 and doing the trial in Angelman syndrome. So we did open up an investigational drug application with the FDA. So this is an open IND open this year. The enrollment for the study has begun, and we're expecting the top-line results in the first half of next year. So the phase 2 study is an open-label study. It's 13 weeks of treatment. It is twice daily dosing at a mg per kg dose, which is an oral liquid formulation. And there is an up-titration period until people get to a target dose. And it's a combination of in-clinic and in-home nursing visits during the period of the study. So 13 weeks of treatment, there is a screening period of 4 to 6 weeks, and then a 2-week follow-up. The primary aim is to evaluate safety and then the pharmacokinetics, how the drug is absorbed and distributed in the body. And then it's also looking at secondarily efficacy measures. So given what we saw in the animal model study, we are looking at a range of outcomes, and all of these things are outcomes that are important for individuals with Angelman syndrome: communication, fine and gross motor issues, behavioral issues, GI, sleep, seizures, and also looking at daily function. And again, our aim with this study, particularly with in addition to the safety, for the efficacy point, and I know that Becky from Ionis also mentioned this, is that the aim is really to inform what will happen in the next trials, the larger trials, and informing what types of things we would bring forward as our outcome measures for those trials. This is a snapshot of the inclusion-exclusion criteria. It's not comprehensive. If you'd like to see more detail, you can see our listing on clinicaltrials.gov. But it is including a range of the pediatric population, so 3 to 17, and then it's also across genetic etiologies. Participants also need to do a screening assessment, do a review of their medical history, as well as some assessments to determine their eligibility before they enter the study during that 4 to 6 week screening period. So we have 3 study sites for this study. All the study sites are in Australia, which is sort of our home base. There's a site in Brisbane, a site in Melbourne, and then a site in Sydney. And currently, all of the sites are open for enrollment. So our current status is enrollment has started, all the sites are open for enrollment, and we're working on completing the study to finish as we target for next year, and we'll report the findings when we have all the enrollment finished in the first half of next year. If you are interested in more details about the study, in addition to clinicaltrials.gov, we also have a study-specific website, which you can see the address here, and that gives you a little bit more detail on each of the different aspects of the study and a little bit more about the background for this study. So with this, I would like to thank very strongly the families who are spending their time now participating in the study, and the kids with Angelman who are investing their time and effort. I also wanted to give a special thank you to FAST Australia, who've been very supportive and helping, particularly our team who's based in Melbourne, with the preparations for the study, as well as being supportive of reaching out to the community. And of course, the FAST global community. I have been involved with the ABON group, and a lot of the development of the design has been, for this study, has been driven by the interactions we've had with the ABON group, as well as with the advocacy groups. So it's really a thank you to all of you, because all the effort you've put in to make all of those resources possible for us. This is a thank you as well for contributing to making this study possible. And thank you for your time, and we look forward to hearing your questions during the panel period.